Hi folks, now we get to focus on the structure of skeletal muscle, really how it's organized. Okay, and so let's get ourselves oriented. We see we have our humerus here, so I'm going to write H for humerus, okay? We definitely have our articular cartilage here, and so that definitely means we probably have a ball and socket joint, not probably. We have a ball and socket joint. That's going to be that glenoid cavity of our shoulder girdle. What else do we see? We see a tendon here. Remember that band of connective tissue. And then we see a hunk of muscle. And so our goal is to go from this hunk of muscle, and then as you can see these little tiny little bundles here, that's going to be a bundle of muscle fibers or muscle cells. And then we're going to take those bundles of muscle cells and pull out one single muscle cell. And as you can see, even those muscle cells can go further and pull out what's called myofibers. But before we get down to that level, we're going to be starting off with the larger levels we see here. And we're going to start from this muscle cell to a bundle of muscle cells to a bundle of those bundles of muscle cells. And they all have their own, of course, terminology. And understanding our larger piece of muscle is going to help us understand, again, the gross anatomy of muscles. But what are muscles comprised of? First, they're made of muscle cells, which are also called muscle fibers. A lot of times you'll see us say fibers, but you could also say muscle cell. And these muscle fibers are going to make up the bulk of the muscle itself. Then you can talk about nerves. And so I'll show you where the nerves are. You see these little yellow pieces here? Those are all nerves. Another piece here. And these nerves are going to connect to each muscle fiber and run all through the muscle. Because remember, we need to be able to contract the muscle. So it has to go through the length of that muscle fiber or muscle cell. And again, we need the nerves to be able to connect to that. Next, we have blood vessels. Now, blood vessels are going to be for every muscle fiber as well. Why? Because we need oxygen. As we're burning ATP to contract the muscles, we're going to need oxygen as well. And that's going to come from our blood vessels. So we have the blue and red blood vessels here. You also see them grouped over here, blue and red as well. And then finally, we have connective tissue. And there's several layers of connective tissue. You'll see that we have these little boxes here that are just showing different types of connective tissue that we'll keep going in through. And also, we already talked about tendons, which are bands of connective tissue as well. So we're going to be putting this all together. Started with this smaller muscle piece that's pulled out. And the smaller muscle piece here is a muscle fiber, individual muscle fiber, also called a muscle cell. Okay, so it's going to be long, the length of whatever that muscle is, multinucleated, like we saw before. It's going to have multiple nuclei, and one muscle fiber is the length of that particular muscle. Now, here's where some connective tissue comes in. The muscle fibers or the muscle cells are surrounded by endomysium. Endo means within, and the mysium is going to tell us within that muscle. So within that muscle cell, muscle fiber, it's going to be surrounded by endomysium. And so we have this whole thing here. You can see this is all endomysium that's surrounding it. Endomysium, endomysium. Now, you might be saying, hey, I pulled out one muscle fiber, a part of this bundle of muscle fibers. So where's the endomysium there? Well, first, let's go ahead and think about what a bundle of muscle fibers are. A bundle of muscle fibers, put that here, is called a fascicle. So first, if I'm looking at this fascicle here, and I pull out one of the muscle fibers, that means that a bundle of these muscle fibers and all the connective tissue you see here is also going to be endomysium. So sometimes you can say endomysium, which is normally what I say, but also sometimes I say endomysium. Okay? Now guess what, folks? A bundle of muscle fibers also has its own connective tissue surrounding it. So if one single muscle fiber and, it's going to be surrounded by endomysium, and a bundle of them internally are going to be surrounded by endomysium, this whole bundle is now surrounded by perimysium or perimysium. Okay? So this whole little piece here is going to be perimysium. And as you can see, when we get to an overall muscle, it's going to be a bundle of fascicles. So if they're all surrounded by perimysium, then all of this here, all this connective tissue here, all the white portions here, is going to be perimysium. Remember, all the blue here is going to be endomysium. So let's review. A single muscle fiber or muscle cell is surrounded by endomysium. When you have a bundle of those muscle cells coming together, that's called a fascicle. And that is formed or surrounded by perimysium. And when you have a bundle of fascicles coming together, that forms a muscle. Okay, so the question is, what is the muscle going to be surrounded by? Well, the muscle is going to be surrounded by epimysium. So this will be all that epimysium. This whole thing that's surrounding this muscle is epimysium. Some layers of detail. We have our muscle fibers, a bundle of muscle fibers with a fascicle, and remember, peri means to surround something. So perimysium means that you're surrounding that bundle of muscle fibers. And usually you're thinking about at least 12 plus muscle fibers that are going to be bound to create a fascicle. Okay, so that's a lot of different muscle cells right there. And also you can go further, and again, you can say that peri means surround to help you remember that surrounding muscle fibers, a bundle of muscle fibers. And then we have our muscles, which is a bundle of fascicles, right? And they're going to be surrounded by epimysium. And so epi, you could just mean, is going to mean like surface or the outside of something. So that will just help you remind you that it's going to be on the outside of the entire muscle itself. How can we make a real world connection? Go to the grocery store, if you got the cash for it, and buy a steak, right? When you look at the steak, you see marbling of the steak. That is going to be that perimysium. Now, sometimes, depending on how fancy the steak is, you can even see some of the perimysium and the endomysium of the individual muscle fibers can kind of coexist and just look like overall larger marbling. That would be like with the fancy Kobe beef, or Kobe beef, whatever you want to call it. Okay? But if you're just going to get a regular steak and you're looking at it, you see all that marbling, that is the perimysium. Okay, so that's going to be, again, those bundles of muscle fibers, and that's what a fascicle is, and all those fascicles are surrounded by that perimysium. I also want to talk about attachments. So remember, we have a lot of connective tissue, and connective tissue can connect things, right? And there's two types of attachment you can see skeletal muscle do with other parts of the body. The less common is going to be a direct attachment. That means this epimysium is going to directly attach to the periosteum, or that connective tissue layer that's surrounding a bone, or the perichondrium, which again is that layer that's surrounding the cartilage, right? And that epimysium can directly attach. And a lot of times we don't see that as much. Direct attachment, okay? That's not something that we see. All right, direct attachment here. Yes, it can happen in the body, but that's not the most common. What we most commonly see and what we kind of are used to are things like tendons, or we'll talk about aponeuroses, okay? Now, tendons we're definitely used to. And what do tendons do? They're going to connect muscle to bone, okay? So remember that. And this type of connection is an indirect connection. So muscles of the muscle connective tissue extend beyond the muscle. You can see here in this perfect example. Let me get a different color. So this muscle, epimysium, is going to extend beyond the actual muscle. And then you can either create a tendon, which we like to have here, or aponeurosis. Now, a tendon is connective tissue connection to bones. That's going to be like rope-like. Okay, we see that this tendon is sort of a rope. It's taking all of that particular epimysium and bringing it up to kind of this little connection point here to indirectly attach the muscle to the bone itself. Again, attaching to our humerus. And we also have sheet-like attachments like aponeuroses. Indirect, because you can think of like the ab muscles, which are flat sheets. And those are aponeuroses, which again, they're muscles, but they have an indirect attachment to things around them. And these connective tissues, our tendons and our aponeuroses, are going to anchor the muscle to the bone or cartilage or also other muscles, right? So again, when we're thinking of our abs, we're talking about aponeuroses, we're thinking of sheets of muscle tissue, okay? Let's go ahead and review again when it comes to connective tissue. Remember, we have endomysium, perimysium, and epimysium. So the endomysium is going to be connected to each muscle fiber cell. That's going to run down the length of the actual muscle. Again, the fascicles are surrounded by perimysium, which is going to run down to the length of the muscle. And we also have epimysium, which is going to run down to the length of the muscle. And they're all going to reach the end of the muscle and be bound together. And they either are going to form indirect attachments in the form of tendons, which are like rope-like, which we see here, or like aponeuroses, which are sheet-like. For us, tendons might be easier to understand because, again, we already talked about joints. But when it comes to aponeuroses, it might be a little bit harder. So in plain terms, it's basically like saying your ab muscles pull on your ribs and your hips through these wide, tough sheets, like flat tendons, okay? And that's where the aponeuroses are going to be. Okay, again, they're indirectly going to be attaching to like your ribs or your hips when it comes to your ab muscles, and they're kind of like flat tendons versus being like these rope-like tendons, if you want to think of it that way. These aponeuroses is that they can actually blend into the periosteum of bone, or they can blend into a nearby muscle fascia. Okay, so just keep all those different things in mind. That's when we talk about the being able to, again, having connective tissue bound together to form those aponeuroses and to form those tendons. We'll talk about the structure of skeletal muscle, but now we must get microscopic. And we're going to actually focus on that muscle fiber, that individual muscle cell. And this is going to help us understand how muscles shorten and therefore contract. But let's go ahead and get oriented. So on our previous visual, we saw that we started off with a muscle, and that muscle was made up of bundles of fascicles, and those bundles of fascicles are made up of bundles of muscle fibers. And that's kind of where we ended. Now we're going to go further and talk about muscle fibers and how they're made up of bundles of myofibers. And those myofibers are made up of repeated units called sarcomeres. And those sarcomeres are made up of myofilaments called proteins. So yes, this is where the microscopic portion comes along. And we learn all of this to help us understand how muscle is going to shorten. So let's go ahead and get oriented with our new visual here. Again, we're looking at a muscle fiber or a muscle cell. And again, that muscle fiber is made up of bundles of myofibers. So even if I go back, remember I told you how you have little bundles here? Those are the individual myofibers. Okay? So muscle cells even have their own bundles. Now, let's go ahead and remember, muscle fibers, are going to be surrounded by endomysium. So we can even see the endomysium here. It's kind of like pulled back. And then we see like a translucent membrane that's surrounding this overall muscle fiber. So let's go ahead and talk about that. That's actually going to be the cell membrane of the muscle fiber. Okay? The muscle fiber is made up of myofibrils, then the cell membrane is wrapping around those myofibers. Okay? So again, you're going to see that cell membrane wrapping around each of those individual myofibers. Now, the cool thing about skeletal muscle, just like every other system we talked about, they have their own little words to help you remember that you're talking about muscle. So first, instead of calling it cell membrane, we're going to call it the sarcolemma. So sarco means flesh, and lemma means to cover. Okay, so the sarcolemma is going to be that cell membrane or plasma membrane wrapping the myofibers. Okay, why is this important? Well, the sarcolemma is going to be there to help send signals to the muscle fiber for it to contract. But guess what? It's not going to be able to send the signal as deep down into these myofibrils as needed. It's going to help to do that. So there's also going to be what's called T-tubules. And these are also part of the membrane, but they're going to be a part of like kind of membrane canals. You can also call them tunnels. And the goal is to reach deep into the myofibrils. Because if I have a signal in the sarcolemma or the plasma membrane, I need to make sure it gets deep down into the actual myofibrils, and then of course cause an actual change in the sarcomeres due to their myofilaments changing. Again, going to this little piece of our little successive visual here. Okay? So let's orient ourselves. We have our endomysium, we have our sarcolemma, and then we have the T-tubules, which are going to help us get that signal down deep within the actual myofiber. And the T-tubules are these little pink portions here. You'll also see them right here. So basically, it's almost like you're going to surround the myofibril like a little ring. That's what we're doing. So far, what do we understand? Say we actually have a stimulus or some type of signal to tell the muscle to contract. Well, the sarcolemma is going to pick up on that first. And then to get deeper down into the myofibrils are, you're going to go into these canals or the T-tubules. What's the purpose of that? It's because we still are not done sending that signal. Now we get to actually stimulate another organelle called the sarcoplasmic reticulum. Reticulum we've seen in just our general understanding of cells. It's an organelle of the cell, okay? Now the sarcoplasmic reticulum is a specialized endoplasmic reticulum of muscle cells. It's going to surround the myofibrils as well. And one of the main things it does is store and release calcium ions. That's going to be helping us be able to, again, get that signal started to contract, okay, or finish that signal to contract. Let's go ahead and orient ourselves of where this sarcoplasmic reticulum will be. See all this blue here, all this blue here. that's going to be sarcoplasmic reticulum. Just like we have with any endoplasmic reticulum, yes, they have all these little areas that are opening, moving things around, and all that's going to help us store calcium. So let's go ahead and review. We have the endomysium that's going to be surrounding the muscle fiber or muscle cell. We have the sarcolemma that's going to be surrounding our myofibrils. And then we have two T-tubules that are going to be surrounding the myofibrils like a little ring. We also have then sarcoplasmic reticulum surround the myofibril. Remember, we're saying they're surrounding the myofibrils, and the myofibrils are just going to be basically the muscle fiber is going to be bundles of those myofibrils coming together. So if you were to just pull and look at all of them kind of this cross section here, you'll see that you have sarcoplasmic reticulum surrounding it. There's your T-tubule rings, and then we also have our cell membrane surrounding it. Let's get a visual of this. You're going to have some kind of signal. It's going to go to the sarcolemma, then it's going to go deep down into the T-tubules, and then the T-tubules are going to stimulate that sarcoplasmic reticulum. When that happens, you're going to have a release of calcium ions, and that's going to allow the release of calcium ions into the myofibrils for contraction. Focus on those myofibrils. These are long rod-shaped organelles. Again, a bundle of them make up the muscle fiber, and they are going to be surrounded by our sarcolemma. Myofibrils are going to contain proteins. They're going to be called myofilaments. Okay, and we know filaments are proteins, and you can even think of these microfilaments, for example, if you're thinking of the cellular structure of the cell, or the cell structure. Now, there's two main proteins we talk about when it comes to myofilaments, and that's going to be actin and myosin. So those are going to be the two main myofilament proteins. Of course, there's other proteins that help, but that's going to be the main ones that we talk about a lot. And these myofilaments are going to make up these contractile units called sarcomeres. And these are the fundamental units of contraction. Okay, so without the sarcomeres, and without the sarcomeres being made up of myofibrils, then you will not have any contraction in your muscles. And this is also what creates that striated appearance that we see in our muscle cells. So let's go ahead and look at this. Again, we have our endomysium, we have our sarcolemma, we have our T-tubules and sarcoplasmic reticulum. We're going to pull out one of those myofibrils. And you'll see and notice that there is a dark area, there's a light area. There's a dark area, there's a light area. You also might even notice little kind of zigzags here. And those zigzags are telling you or giving you boundaries of one single sarcomere. So I can see a zigzag here and here. That means that that's one single sarcomere there. And what is it made of? It's made of myofilaments. What are those two myofilaments? Actin and myosin. So it's going to be the myosin that you see in purple here, and the actin is going to be in the light portions. So when you're thinking of striations, and we can go back to that as well. Let's go ahead and go back up here. And you see that we were talking about striations in the previous video. There are lighter portions, darker portions, lighter portions, darker portions. That is going to be sarcomeres. Okay? Take this little sarcomere here and blow it up here. As you can see our actin filament. So this is going to be actin. You can see our purple myosin here. Myosin. You can also see the zigzag line. And we'll get all the different labeling as we go to the next video. But those zigzag lines are giving us the boundaries of that individual sarcomere unit. And as you can see, that myofibril is made up of multiple repeating units of sarcomeres. And it's inside those sarcomeres that you're going to have the myofilaments. Okay? Now, you also are going to see things like the M line here, the Z disc, and some folks say, why is it zigzag? Because remember, we're talking about cylinders and cross sections. So when you're seeing them in kind of like this 2D format, yes, they look like a zigzag. We're also going to talk about the elastin here, elastic protein. We're going to talk about this zoning area that has a lot of space. We're also going to talk about how the actin is going to begin at this Z disc area and end where the myosin ends here. So let's get into that in the next video, and I'll see you there.